This is Audiomunity, a podcast about our body's never-ending fight with the outside world. Hello, this is Audiomunity, episode 53, recorded on March 5th, 2026. I'm Kevin Bonham at Tufts in Boston, joined as always by Matt Woodruff down in Atlanta.
Hey,
Hi, Matt.
everybody. How's it going? Kevin, I have a bone to pick with you. Before
Tell
we
me.
start, I was looking back through our last episode, and I've been involved in some stuff, and time has been thin recently.
Yes.
And during the
It's
last
March,
episode,
and we've only recorded 2 episodes this year.
That's true, and I apologize for that. That is all on me. Can't guarantee that that's going to change drastically because of the way the world is right now. We'll get to that in a second. But
How is the world now, Matt?
not great. But I was looking back at that episode, and I looked ridiculous.
Yeah.
I looked ridiculous. My
didn't want to
beard
say anything.
was like out to here. Like I looked, it was horrible. So
Like a
I
mountain man that
apologize.
came down from... Yeah. I wasn't going to say anything, because it looked
Well-
like you were just slowly growing, like, resistance beard.
I mean,
we
I
have
was.
thesis beard. Probably
Yeah.
eventually you will have tenure beard. Not that I can participate in any of this, as you know.
Right. Well, we tried that.
But yeah, so we're doing the best we can here. Part of it, we recently made a shift to Substack. You can find us now on audiomunity.substack.com, or is it substack.com/audiomunity? One of those things.
Yeah, Google will get you there.
Audiomunity.org is still the home base. There's links there to everything else. Eventually, that's going to redirect to Substack. We still need to figure that out. But
All
we're
the
just
links.
going. We're in the midst of stuff. We had a giant snowstorm here dump like ungodly amounts of snow on us. School out for 2 days after February vacation. So we had the kids at home for 9 days straight, 11 days straight. They were freaking out. Samara is potty trained now. All kinds of good stuff happening on the personal front. But Matt, what's good in science in 2026?
Not much. Hasn't improved much since 2025. And yeah, I mean, yeah, it's just not going well, Kevin. But I think one of the things that we wanted to talk about was this NIH grant review restructuring, and this is something actually that we've touched on previously because there have been kind of iterations of this that have been sort of slowly implemented, and we've talked about it, as these red flags have been raised around what happens when you take grant review, peer review, and you sort of allow that to happen, right? You allow peers to review each other's work, but then you add an additional layer of bureaucracy on top of that initial grant review, right? Like, imagine if you were, for example, to try to control the kinds of science that is done at a large geographic level, let's say at the level of a country. What you might do is you might keep a lot of the lower institutional practices in place, but then say at the end of all of that, and then we'll review everything that's about to get funded to make sure that it goes into the goal.
Let me make an analogy here, because this came up recently at home, where we went and we asked our 2 kids, 7 and 2.5, we said, let's have a vote on what we should have for dinner. And everybody put in their vote, and then we just decided we're going to have the dinner that we had planned anyway. And that's very much what seems to be happening, although it's a little opaque. And I think it might be worth talking about, because, to be honest, I was mostly ignorant of all of this stuff before, like, 12 months ago when I started this position. Like, I understand you send grants to the NIH, you get reviews back, but I didn't really understand how it works. I think I have a little bit better understanding of it now, although I still haven't been on any study sections, so I haven't been part of the peer review process. But I've now submitted a few grants, gotten some reviews, and done a lot of reading. And so the way it used to work, or the way that it often works is that you have within the NIH, and I think NSF has similar structures, although they're slightly different in the details. But basically, at NIH, National Institutes of Health, you have a bunch of different, they're called institutes and centers, ICs, that have different focuses in terms of their research. So you have NIAID is the National Institute for Allergy and Infectious Disease. You have NICHD, which is National Institute for Child and Human Development, Child Health and Human Development, I think. You have, the Lung Institute, you have the Cancer Institute, all of these different things. When you submit an NIH grant, like an R01, you send it to one of these ICs typically. Within those NICs, they have specialized study sections that have experts of various kinds.
You do that because, because they know what they're talking about, and they know what they're talking about.
So if you're studying cancer, you don't want to be reviewed by people who study insect migration. You want it to be studied by oncologists and people who study cancer, right? So there's a little bit of strategic, like some institutes have more money than others. Like there's a little bit of strategy there, but mostly it's like you submit to the institute that has the right expertise. Within the institute, there are study sections that have even more specialized expertise. So if you're studying cancer, you might have a study section that is focused on like chemotherapeutics. I don't actually know what's within NCI, but maybe you have a group that's like on chemotherapeutics. Maybe you have like a melanoma specific study section. Maybe and those study sections have folks at the NIH that are sort of career civil servant scientists whose job it is to shepherd these processes. You have program officers that manage grants. And then those study sections recruit scientists like us who have previously been funded by NIH and have the relevant expertise to come in and actually review grants. There's a whole bunch of criteria for what you score in terms of grant review, and it's for impact, it's for feasibility, it's for environment, all kinds of stuff. Those scores get aggregated, and they're given essentially a percentile rank. So you score each of these things, you average across a few reviewers. Then certain people are like deep diving on your particular proposal and a small number of others. You have for the proposals that score well from those reviewers, it goes to the whole study section. There's a discussion between the people who did a deep dive. Other folks probably just read the specific aims and maybe skimmed the approach, but there's a discussion. Everybody votes. You get an aggregate score. That score for many institutes would give you a percentile rank. And it wasn't true at all institutes, but for most institutes, there was basically a pay line where it's like any grant that scores lower than some value, usually in terms of percentile. So any grant that is in the like 8th percentile or below is going to get funded. And then there's a little bit left over for people that scored like, if the pay line is 8%, people that scored like 9 or 10, they might get a second look. And if their grant was really like well aligned with the institute's priorities, those might get funded.
That's where the politics of all this comes in, by the way. Right? Because in addition to, writing a really good proposal, in addition to having impactful science, in addition to all of those things, and this is both just an important part of how human beings work and also sometimes a knock on the way that this process plays out. If you know the program officers well, if you have proactive discussions, if you sort of engage in the process, if you sit on a bunch of study sections that are important, if they know you, right? These are all things that might take your grant from an 8th percentile funding line to a 12th or a 15th percentile funding line if you're really doing a lot of work, right? So there is a human component here, just inherent in the fact that these are human beings. But by and large, what we're trying to do here is we're trying to get a reasonable assessment of each grant that comes into the door by a group of scientists in that field that can realistically give it a score.
And realistic and as best as is possible in this kind of thing, objective, right? That's the other thing, is that you were trying to get some consensus from a bunch of scientists. And often you have like, I just had an R21 reviewed, and it was like reviewer 1 scored super well, totally understood it, was super on board. Reviewer 2 clearly had no idea what the grant was about, scored it terribly. Reviewer 3, like, mostly understood it, had some quibbles, and so they were sort of at like a middling rank. And so like all the comments are addressable, we're going to resubmit, so all of that. But like it is as objective within the framework of people being humans and not everybody having the same, ideas about what's important. It's as objective as possible. And basically, so it's also worth noting that not every IC does it this way. Not every study section works the same way. So some institutes have never had pay lines or haven't in a long time, and that's fine when you expect that the people making decisions up and down the ranks are scientists that have the interests of the scientific community and the interests of innovation and research well in mind. You can say like, well, we're going to take even, so anything that scores 20% and below is an excellent grant that should be funded. The reason pay lines are so low is because there's not enough money to fund everything, right? So I think it's totally reasonable to say, like, we're going to say anybody that's 20% and below, that goes to the program officer, that goes to the broader institute to decide which things are going to be priorities, either because like it's a new PI and we want to foster innovation in young scientists, or because like this project in particular is well aligned. Like that totally makes sense. But what's happening now is that pay lines have been eliminated across the NIH. There's clear language being put in that says the institute directors get the final say on what gets funded. And it is very clear from all of the other moves, it's not just those specific policies, but it's all of the other stuff around that is basically saying what's actually happening is that we're going to have the veneer of peer review, and then the administration is just going to fund the people that they want to fund. And they're going to axe the people they want to axe. If you said the wrong thing on Blue Sky or on a podcast, maybe, we don't know. Like, these decisions are non-reviewable. They don't have to give justification. They basically get to fund or, crucially, not fund whoever they want. And it is completely unreviewable, completely opaque, no transparency about why. And if this was a normal administration that hadn't done all the other bullshit, you could say, like, this is just a different policy and we think it's going to happen in good faith and all of that's fine. But that's not the world we live in. And basically, we're now having Lysenkoism is basically what we're going into. We're basically going to have the administration fund the people they want to fund, use grants as bargaining chips and as punishment and reward for cronies. And yeah,
I was trying to come up with the word to the transformation to Politburo style of grant review. It's really hard to say Politbureautization, but I think that's the word that I would like to start introducing.
Politbureautization? Okay.
Yeah, Politbureautization.
I can get behind that. We'll see how the transcript figures that one out to the AI trying to transcribe this episode.
No, it's fine. They have me for so many other things at this point. So I recall back to the beginning of all of this. One of our earliest What's Good in Science 2025s, I recall a conversation that you and I had where family members, like, we would talk about this stuff because the trajectory on this has been in retrospect now clear for a very long time. And I think that, it's always hard to put a hard point on it in the moment because you don't know if it's a blip. You don't know if the trajectory is real. Like, you can see the trend, but you don't know where it's going to fall. And I remember us talking about the frustration in being generally pretty reasonable people. I'd like to believe that we're generally pretty reasonable people. I'd like to believe that we can have conversations with people that we disagree with. I'd like to believe that we're mostly respectful and open to new ideas. And it is a strange place to be that person and also telling people that something is about to happen that is absolutely catastrophic. And it's sort of out of line with personality and discussion styles, I think. And I think it was you that pointed out that to be the sky is falling radical in a position in a family setting, probably for the first time, is a really jarring place to be, because you're like, hey guys, like, I know that this is arcane, but this actually could get really bad really fast, right? And everybody's inherent response is like, I mean, it couldn't be that bad, right? Mm-hmm. And I would just like to say, starting off What's Good in Science 2026, we did it. We got there.
Yeah, it got that bad.
We are
well,
almost fully.
Here's the thing. So, yeah. I mean, the problem is this stuff will not be visible.
Yeah. Well, that's
even from within science, like, labs that probably should have been funded aren't going to get funded. Labs that probably shouldn't have been funded probably are going to get funded. But those decisions, like, for most scientists most of the time, that's already been, like, when I was in grad school or when I was a postdoc, I didn't have any insight into which labs were getting funded. I knew that some were very successful and some weren't. But, like, why that was happening wasn't necessarily clear to me. And like, most of the time, scores and reviews are not made public. Like, you can see in NIH Reporter what grants get funded. You can see a little blurb on what they study, but you're not seeing all the details of those grants. So it's going to be very hard to quantify the effect that this is going to have. And again, because, you know, not every IC had pay lines before, people are going to say, well, you weren't objecting before when Institute so-and-so didn't have a pay line, so why is it a problem? And, like, the problem here is that the effects of this are going to be invisible. By the time they're visible, it won't be clear what the cause was, right? So in 10 years, when we don't have a treatment for X disease, when we don't have all of the vaccine thing, like, that's a little more visible, but, like, all of the innovation that doesn't happen, all of the labs that close and the people that go into pharma or completely change careers, all of the, like, pipeline effects of trainees that don't get trained, people that don't get PhDs, people that go on to become TikTok influencers instead of researchers, like, those effects, we will not be able to trace it back to this particular decision. But that's going to be the effect, is that a lot of US science is going to not happen, which means we're not going to get the innovation and the advancement. We're going to lose a huge amount of workforce, and shit's going to suck, is basically what it comes down to.
Yep. It's bad.
The good news, if there is good news,
every upper respiratory infection.
We did. We have now,
yes,
based on the paper that we're going to read today, we've eliminated infectious disease as a thing. So maybe we don't need all of this training anymore. We just don't. Yeah.
No,
all it's going to take is that we just need to have the symptoms of a cold for our entire lives, but then we'll never get infected. So, you know.
Okay. So
everything will be great.
For those of you following along, Kevin's about to jump hard onto the snark train, and that's fine.
No.
But let me give you some red flags about this particular discussion. And maybe put some guide rails on it. So the paper that we're about to talk about is a science paper. It's actually pretty fascinating, but it's a science paper titled "Mucosal Vaccination in Mice Provides Protection from Diverse Respiratory Threats." The first author is Haibo Zhang. Last author is Bali Palendran. So, couple points of note, little context. I trained with Bali Palendran. I like Bali a lot. I think he's a great scientist. I do not know Haibo Zhang. Mehul Suthar is the second to last author on this paper. He is in the Emory Vaccine Center. I've worked with him a lot. We're collaborating. I know many of the authors on this author list.
Ralph Barrick is another like giant in the field. So yeah,
exactly. So we've got a paper from some well-established people here coming out of Stanford. I actually think that before we get into the reality of like how we see where this paper fits within immunology, I'll start by saying it's good work, right?
100%.
So none of what we're about to talk about, I think, is actually going to go to the quality of the science here. I mean, Bali is an extraordinarily thorough PI. I've worked in his lab. Going through this, there's really nothing to be said. Well, we should go through it briefly just to talk about, what the actual experimental designs and things were. But I honestly don't think that that's where we'll spend the majority of our conversation today.
No, the science in this paper is solid.
Yeah,
all the experiments are solid. Yeah. They do the things that I would have done in the same place. I mean, to the extent that I would have any idea what to do in this field, everything makes sense to me as I read it. It's like, oh yeah, that's the right experiment to do next. So,
yeah, and I think it's funny, and I'd be interested, and I may reach out to see, but I almost wonder in reading this paper if each subsequent figure was like, but this won't work, will it? And then it does. And it's like, but this won't work, will it? And then it does, right? So let's just start with figure 1, because I think this sets the stage, and I really like a solid figure 1A that's like, hey, I'm about to show you a science paper, right?
But before we do a figure 1 though, I want to hit one point in the introduction, which I didn't know, which is kind of fascinating, which is apparently this has been known for some time, that you can, like, when you vaccinate against certain things, you will observe, especially if you do intravenous vaccination, you will observe broad-based protection against other pathogens. So they talk about this in the context of BCG, which is a vaccine for tuberculosis. They talk about this in terms of oral polio vaccine. So you have these other vaccines that when you get them, you get a transient protection from other things, which honestly, like it makes sense, but I didn't really know that this was like a clear effect of certain vaccines that we often give to people. So I think that's crucial background going into this.
It is, and I was actually going to return back to that background after 1A, but let's, since you introduced it, let's talk about BCG vaccine for a second. So BCG vaccine is against tuberculosis. It is I believe it's a live attenuated, it's a live attenuated Mycobacterium bovis, right? And the BCG vaccine is really effective against a specific kind of tuberculosis. It's really effective against severe childhood tuberculosis, right? It is sort of variable in terms of adult tuberculosis. The numbers I got back are like 0 to 80%. Like it's a wide range, but it is highly effective in places where tuberculosis is endemic at protecting children from its most severe effects. Which means that in places where tuberculosis is endemic, people tend to get vaccinated with this. So that is the primary use of BCG, right? Here's an interesting secondary use. In the 70s, people realized that you could take BCG vaccine and you could insert it directly into the bladder via a catheter for patients with bladder cancer. And the result of that is that a local immune response will be mounted and will attack the cancer. Because the BCG vaccine is highly immunostimulatory, right? And so what you're actually talking about there is immunotherapy, cancer immunotherapy, right? You're creating an inflammatory, stimulatory, or stimulating environment directly in the bladder around the tumor. And presumably, and we've talked about this a lot, right? Tumors which rely on suppressing immune responses in the local environment fail to suppress that. And so your immune system picks up on it, right? We've talked a lot about how the adaptive system very much can respond to a tumor, even though it's selfish, right? You just need the right brakes to be pulled off. And so this is an example where you can use the non-specific inflammatory effects of a therapy like BCG in order to target something completely different, right? And I would say that that became standard of care, right? So since the 70s, we've been using BCG vaccine as standard of care for treating bladder cancer, right? This is not a new phenomenon that we're sort of looking at. And I would say, during COVID, one of the things that has been known, again, in communities, in scientific communities where this is routinely administered, is that there are knock-on effects of systemically administering these kinds of things. There were a lot of studies in Russia, and I think also in China, when COVID-19 came around, asking the question, if we vaccinate or if we boost people with BCG, is that going to prevent them from the most severe versions of SARS-CoV-2, right? So this is an idea that has been batted around for more than a century now. I think that BCG is more than a century old. It has been used in a number of different modalities. There's always this kind of question mark as to what you're actually studying because it's just so complicated, right? You're essentially using a system-wide inflammatory response in order to prevent an incoming pathogen or, a breaking of immune tolerance. So it's a really fascinating history, actually. It's complex immunology. It's really cool work. So I think the fundamental question of this paper is like, but why though? Right? So yep. if you're asking, how do we create broad protection? It seems like there's some signature of these systemic BCG vaccine responses that do provide broad protection. And I don't know, Kevin, if you remember this, but the reason that I went to Bali's lab to begin with was fundamentally because they published a paper while we were still in grad school. So this was back in the 2010s, 2012s or something. And Bali came to Harvard and he gave a talk. And I thought it was a fascinating paper. And basically what it was about TLR combinatorics. And basically, how do you combine different toll-like receptor signals? And in those different combinations, how are you polarizing dendritic cells, I think the paper was on at the time, and how do those responses differ depending on their polarization? I don't remember the details of the paper, but it was really cool. And I was like, well, that makes a lot of sense. You've got different toll-like receptor signaling pathways, presumably in complex environments. The immune system is going to do different things with them, right?
Yeah, and fundamentally, it makes sense from an evolutionary perspective that we have lots of different ways of detecting microbes. We have lots of different ways of potentially responding to microbes, and we would want to align the specific type of immune response to the specific type of infection. Absolutely. Whether that's what kind of organism, is it a virus or is it a bacteria? Is it extracellular versus intracellular? Is it a fungus? Is it a worm? All of those things are going to have, we don't get infections with pure LPS. We get infections with microbes that have all kinds of different stuff going on. And we want to have redundancy and we want to have polarization that's going to target a specific thing. When we're designing vaccines, for a long time, the best vaccines, well, for a long time, the only vaccines, but even in more modern times, the best vaccines are the ones that mimic real infections because you are getting the immune system to drive in the direction that is most effective for treating that specific kind of infection.
Yeah.
And so if we could figure out the fundamental units of polarization, which combinations are driving at different immune responses, we could really tailor the responses of the vaccines we produce to be the most beneficial and provide the best protection.
Yep. Yes to all of that. So it also stands to reason that you might be able to take those signals in combination and create a version of something that looks like BCG, create that large systemic response and ask the question, can you get the same kind of broad protection in a less chaotic, live attenuated bug, less controlled environment, right? So that's what they did. So figure 1A is essentially a very, very classic immune model, right? Which is ovalbumin. And I love ovalbumin. It is a protein that is highly abundant in chicken egg whites. It's been used as a model vaccine antigen for a very long time. So this is a way that people study immune responses. It's a way that I've studied immune responses in the past. And essentially, they're doing something very simple here. They're taking ovalbumin, a protein antigen, and they're coupling it with a TLR7/8 agonist and a TLR4 agonist, right? And those are important because those are different TLRs that go down different signaling cascades, right? You know this better than most people-ish. Okay, I'm getting the... But regardless, yes, they
TLR4 typically responds to LPS, which is from the outer surface of bacterial outer membranes for gram-negative bacteria, and TLR7 and 8 respond to nucleic acid inside of endosomes.
Yeah, so you got different compartments that you're signaling from, right? You're increasing the complexity of where those signals are coming from. So literally what they're doing here is they are vaccinating 4 times. Okay, so they're taking ovalbumin and they're just injecting mice. And when I say injecting mice, what I mean is through intranasal administration. Okay, so they're not, as far as I can tell, aerosolizing, right? What they're doing is probably applying to those upper airways dropwise. And let me actually go through the methods.
That's a good point. I didn't actually pay attention. I assumed they were spritzing it.
So these antigens are being administered, or these immunizations are being done intranasally, and it's no more complicated than taking 20 microliters of the formulation that you've come up with and basically just dropping it into the nostril and allowing the animal, under anesthesia, of course, allowing the animal to inhale that 20 microliters, right? And so what you do there functionally is you inoculate the upper airway, right, as you would with a person that just inhaled a bunch of stuff up their nose, right? You're going to get into the lungs a little bit, but it's really shallow lungs. You're not going to get into the deep lungs. It really is kind of an upper airway and nasal passage administration here. So what they're doing is they're adding these things in dropwise. And again, this is ovalbumin, right? This is not a viral protein. It's not a bacterial protein. It's nothing, right? It is a small foreign protein with the right mix of TLR agonists. And then in figure 1B, they infect those mice with SARS-CoV-2, a mouse-adapted variant of SARS-CoV-2. And they do this in 4 different contexts. Obviously, one unvaccinated, one that has been given the OVA vaccination 21 days post-vaccination, or giving the virus 21 days post-vaccination, one that's been given 42, and one that's been given 3 months after. So this is 90 days. And what they show fundamentally is in all 3 of those settings, out to 90 days, there is a significant reduction in weight loss, which fundamentally equates almost directly to mortality, because weight loss at a certain percentage, you have to sack the mice, on and on. So what they're basically showing there is they can take an irrelevant protein, they can immunize the mucosa of these animals a number of times repeatedly, and then wait 3 months and come back with a lethal dose of SARS-CoV-2, and those mice are protected. And that's surprising. So then they look in the lungs and they look for differences in pathology, and sure enough, there are clear differences in pathology. The lungs look a lot more open and a lot more normal in the mice that were immunized. So then, of course, they do the, I think, natural follow-up experiment, which is like, okay, well, let's do it with a bacteria or some other pathogen. They do the same thing with staph, and they do the same thing again with a third pathogen that I don't know much about.
It's sort of an emerging bacterial pathogen. It's called Acinetobacter baumannii. It's kind of cool, but yeah.
Cool. All right. But regardless, in both of those bacterial pathogens, the same thing holds true, right? So they're basically looking for the colony forming units, right? The amount of bacteria in these animals essentially after inoculation. And the answer is that in animals that have been primed with this completely independent model
irrelevant
antigen with the right TLR signals, those bacteria essentially do not take hold.
Yep.
Right?
Yep, it's bonkers. It is worth
that's
noting.
Figure one.
That's figure one. I mean, yeah. It is worth noting that you are getting a pretty substantial drop off in effectiveness over time. So as you said, you're immunizing 4 times.
You are
1 week apart. The first version is you are basically infecting them at the same time as you're doing that 4th immunization. That has the strongest response. 3 weeks after that, you are still pretty substantially protected. At 3 months, you are protected, but not nearly as much. So it's declining over time. This is not a memory response. This isn't true vaccination. It's a little weird to say vaccination in the title to me, because effectively you are doing immune stimulation that is long lasting.
Yeah. I would say you could, in other contexts, you could, a flu mist, for example, is not so dissimilar to this, right? And I think in that case, you are immunizing, you are vaccinating, but what you're doing is you're vaccinating against something completely irrelevant, right? You're using a protein antigen to essentially generate an inflammatory response, right? So it's actually not the vaccination component necessarily that is the important part here. It's the surrounding inflammatory context. So and they get into that in figure 2, right? So the first thing that you'd of course ask is well, what does it look like in the lung, right? What are the immune cells doing when you do this, when you go through this for administration course? And the answer is as you might expect early on, but potentially more surprising later. You get a lot of activation of the innate system, right? So they're looking at alveolar macrophages, they're looking at dendritic cells, both CD103 positive, CD11b positive DCs, roughly equivalent to CDC1s and 2s. And so, the first result that's a little surprising is that these systems seem to be activated not just for the time that the immunization is ongoing, right? Because we would expect that to look like a 2 or 3 week response. Really, what they're seeing is a persistent activation of those populations, really all the way out to 90 days. Now, you can see certainly a drop off in signal between that day 21 and day 90. It's not like it is just on forever, but it's still there. So then, of course,
it's substantial. It's clear. It's T cells come up as well. They're activated.
Yeah,
producing
interferon gamma-producing
cytokines.
T cells.
Yeah.
Yep, exactly. So we are getting a substantial innate and adaptive response that are persisting out to 90 days. And in addition to that, they get into some epigenetics, right? Because you start to come to a place, right? When you're talking about 90 days, you start to come to a place, well, this should really have resolved probably, right? So are we looking at short-term changes, or are we looking at, actual epigenetic long-term remodeling kinds of changes? And they do actually see some evidence of epigenetic changes in those alveolar macrophages specifically that they're pulling out, which I think is really interesting, right? So you've got at least a temporary remodeling of how the genomic material in those cells are going to work. Which of course begs the question, well, what part of the system is important then, right? Mm-hmm. So you've got this weird I'm going to prevent everything response going on. You've got activation of the innate system, activation of the adaptive system, innate changes leading towards the epigenetic interpretation, right? are the adaptive systems really important at all, or is this an innate driven response?
Right.
And so they do what is really a simple set of experiments, which is great.
I just want to clarify, like, the reason that you would ask that question is because, again, we normally think of the adaptive immune system as being antigen specific. Like, the thing that matters about T cells and B cells is that they are antigen specific. That's what makes them adaptive. But what we're seeing is that the specificity of the T cells is irrelevant to the pathogens that this response is affecting, right? We're immunizing against chickens, and we're getting responses to SARS-CoV-2, and I think they looked at flu too, right? They did PR8. It's not in the figure, but and then to 2 different microbes that are like totally unrelated to each other. So you would reasonably ask the question: does the antigen specificity, does the adaptive immune system even mean anything, or is it just coming along for the ride?
Yeah, and the answer is it does still. And this is a really interesting finding. I think actually this is probably, I would argue, one of the more interesting basic immunology findings here, which is that if you get rid of CD4 and CD8 T cells, and you can do that pretty easily by just injecting literally an anti-CD4 monoclonal antibody, probably an IgG1, so that complement fixes, so that it dies, right? And then it gets pulled up, or an anti-CD8, or more impressively, both. That response crashes, or the protection crashes, which tells you that even though the response here, the adaptive response, is not specific to the thing itself, right? Even though we're vaccinating against chickens and then challenging with some other pathogen, the T cell response is still critical, right? And this is actually an interesting field, right? This suggests that there is a resident memory T cell population, essentially, that even in an antigen non-specific way is capable of modeling downstream, or is capable of modulating downstream impact, right? So you're almost arguing for a priming environment kind of adaptive response. It would be interesting if at 45 days you eliminated those T cell responses in the lungs, for example, to see if then you lost the protection, or if it's just the
this
early
is something
phase.
that I was wondering. So their treatment, they are basically obliterating the T cells throughout the vaccination course, starting from before the first vaccination. So they're saying you need it, if you eliminate T cells completely, innate response alone is not sufficient to drive this response, which also makes sense. It would be very cool, I think, to say, what happens if we obliterate all of the T cells at the end of all of the vaccination course? Do they continue to be needed for that long-term protection? Or are they setting up the environment and then they're dispensable? They don't really get into that.
No, they don't. And I don't think they absolutely need to. I think it's just an interesting immunology question, right?
Yeah,
whether
totally.
the priming effect is the important part, or whether the persistence of those cells in the environment, because that would make some sense, right? Then what you're doing is you're tracking, a relatively short-lived adaptive system who is able to confer some broad protection in addition to some specific protection, right? But
I think there's also the bit that's like all of these are interesting philosophical things, or theoretical things. We don't have to again, it's not a limitation on the paper, but
not at all.
Another question would be if day 7 you did ova, day 14 you did a hapten, day 21 you did a different thing. Is it that you are repeatedly re-stimulating the same population of T cells that is expanding the cytokine environment to really to just drive all of that cytokine production, which is modifying the innate response? Or is it just any non-specific T cell response is going to get you the right cytokines? But the fact that they have to do it 4 times, to me, suggests that what you're doing is you're you're amplifying those specific T cells over and over and over again in a way that that builds up the environment, the cytokine environment, to make these innate cells really responsive.
Yeah. No, I think that's fair. So now we've got a situation where we know that the T cells at least are important. Reasonable to ask the question, are the macrophages important? Although to be honest with you, and I'll get into this in a second, this is the figure. There's always a figure that I'm not sure needed to find its way in, but I'm sure that they were looking for mechanism, right, and sort of the initiation of the process. So basically, what they're doing now in Figure 4 is they're asking how important are the macrophages that we just identified all those epigenetic changes in, and why, right? Like what is it that could be different? And they use this tool called collagenate-loaded liposomes. And collagenate-loaded liposomes, Kevin knows I have a sordid history with, which is probably why
we've been through it, on autoimmunity even too.
Yeah. Collagenate-loaded liposomes are an interesting tool in that they kill macrophages, and specifically they kill macrophages that are highly phagocytic, and that's because they actually kill anything that picks them up in large quantities. So if you put enough CLL in a cell, the cell dies. So whether you're a B cell or a macrophage, if you can pick up CLL, you're going to die, right? So it tends to hit highly phagocytic populations. It has a lot of bystander effects. One of those bystander effects is it's actually itself an adjuvant. So if you were to take OVA and inject OVA alongside CLL, what you'd find is that you'd actually get an adjuvanted response to the OVA, right? Because there's an inherent inflammatory process to what CLL does.
Yeah, because you're not killing the macrophages in a nice apoptotic, quiet little suicide way. You're exploding them, and they
blow up.
It's
an imaging nightmare. When
freak
you use
everybody
CLL.
else out.
Yeah, and it's fine. So here, what they're asking is, if you blow up all of the alveolar macrophages, can you still control the burden of staph in the lung? And the answer is no, not really, which isn't super surprising because, that's what alveolar macrophages are sort of for, is eating all the stuff that comes into the lung. It's fine, right?
Yeah, I would have liked this experiment more. Well, so there's sort of two things about it. So one is, if you didn't see an effect of killing the macrophages, you wouldn't be convinced that it was because macrophages weren't important. You could argue well, it's just activating everything else, and so that's part of the reason. So seeing that when you kill off the macrophages in this explosive way, you lose protection is good evidence that, yes, indeed, macrophages are important for controlling staph. I would have liked to see this with one of the viruses because macrophages are less important for an antiviral response in general. So it would have been nice to see you're not killing the most effective cell for clearing this pathogen in the first place.
Yeah, and they also don't, and maybe this was just a time limitation, they also don't show the same protection assays that they show throughout the rest of the paper. And my guess is that my guess is that they did that. And they found that protection was fine because collagenate-loaded liposomes will also give you that activated scenario. But who knows? It could be that experiment wasn't done. It doesn't detract at all from the rest of it. But what they do show is that by vaccinating mice in this way, you basically can pull alveolar macrophages, and you can show that they are increasingly active, right? Their phagocytic capacity does go up. So, those epigenetic changes that we identified before do seem to have some phenotype effect. Do I think that phagocytosis is the primary mechanism of how this is working. I do not. I think probably there are lots of things that that those macrophages could likely be doing in that environment. Phagocytosis could certainly be one, and maybe, anyway. I think it's fine. They definitely show that, there is an impact on macrophages and that those macrophages change in phenotype in response to the vaccination. That's probably important in the general establishment of this response, which they get into in figure 5, which is functionally what you're getting by doing this is you're getting the formation of tertiary lymphoid organs. Right. So they do some interesting spatial transcriptomics where they basically show these giant clusters of B cells and T cells and macrophage, like all of those sorts of, all the things that you'd expect for an active tertiary lymphoid environment to have formed. So this, I think, is much more important to me than figure 4, which is that, yes, all of these cell types have individual roles, but what you're really doing here is you are establishing a fully functioning independent immune structure within the lungs by doing this kind of vaccination approach.
Have we talked about tertiary lymphoid structures before?
I think that I don't know have, but let's go into them
just briefly.
Let's go into them just for a second. So most of what we talk about when we talk about adaptive immune responses are confined to secondary lymphoid organs, right? So the spleen and the lymph nodes, and those are areas where lots of lymph
and Peyer's patches
of the body are draining, and it all comes together. And Peyer's patches. And so these are places where antigen is concentrated from peripheral tissue, and when it's concentrated, the adaptive system has an opportunity to kind of see what's going on all around the body. And it has a chance to sort of interpret all of the signals available to it and respond, and then send the adaptive system out to where it's needed in the periphery, right? So
and crucially, those secondary lymphoid tissues they're developmentally regulated, right? So primary lymphoid is sites of immune primary lymphoid development. So like bone marrow and thymus are the primary lymphoid organs. Secondary lymph, is that right? You looked at me skeptically.
No. Okay. I'm sorry. I was looking at the paper.
Lymph nodes and spleen, those develop even in the absence of infection. You have germ-free mice, they will have lymph nodes and spleens. Those are sort of developmentally driven structures that are places where we expect immune responses to occur. Tertiary lymphoid structures are like new aggregates of immune stimulation that are like trying to get a secondary lymphoid structure like environment out in the periphery somewhere where you wouldn't normally expect it to happen.
Yeah, and it's
so that's why seeing these in the lung is the weird thing. It's like you get a lymph node in your lung temporarily that has all of the support structures for T cells and B cells to come together and get all of that secondary activation that they need.
Yeah, so the word you used there was important, the temporary word. These are sort of designed as like flex environments where you can establish something local at the site of infection that's capable of generating a local response and taking advantage of that, right? You see it in the lung, obviously, with severe infection. You see it in the kidney in patients with lupus, for example, with advanced autoreactivity, right? You can see it in lots of different places where you've got these sort of local inflammatory responses trying to control an active and frequently chronic infection, right? These are particularly where you find tertiary lymphoid structures, is where you have chronic inflammation and the failure to clear essentially a pathogen, which is very similar really to what they're doing with that vaccination scheme, right? By introducing it over and over and over again, what you've got is a system that probably feels like it can't actually clear the environment.
Right. And
so
this is, your work has shown that these environments are fundamentally disordered. They are like slipshod environments that do not have, like, they're trying to get that secondary lymphoid support because it's critical, But because they're a little bit haphazard and thrown together, they don't have the exquisite coordination and regulation, and so you end up getting some potentially pathologic responses. I think evolutionarily the idea is like you do this when you have no other choice. And
I would say you do this when there is a local response that is clearly necessary. It's not obvious how the system is going to clear it, right? And so you sort of set up shop closer to home base functionally. So you see this
to
in
try
TB.
more shit out, right? Like
Yeah,
you're
exactly.
trying.
You see this in TB. You see some organization around cancers that are trying to be responded against. These kinds of things, right? So you're just moving home base closer to the site of infection. So you get tertiary lymphoid structures, which is a sort of big deal finding. And then this last bit is wild. So I'm going to just move into figures. So we've talked about pathogens, right? What we're really talking about here is we're talking about long-term polarization, right? The whole paper is talking about long-term polarization of a response in the lung that's actually preventing other pathogens from establishing themselves, and you're basically pre-kicking off a robust response capable of higher antigen presentation, better co-stimulation, right? Like all of those things. And then they go back and they actually do a really fun, like classic immunology experiment. Like if we drive hard a Th1 response, right? Hard, which we've done the whole paper. And then we try to give this mouse an allergy. Can we do it? And the answer is nope. So an interesting thing about allergy, and we've talked about this before, is allergy is highly dependent on a completely opposite and mutually exclusive set of polarization signals, right? So you can't actually take an allergic reaction and at the same time have like a strong, hard Th1. As far as we know, right? These things are pretty mutually exclusive unless they're geographically or temporally separated. So basically what they're showing is that if you do this work and you sort of establish this longer term tertiary lymphoid structure, Th1, interferon gamma driven response, you actually really can't force the mouse into allergy because it doesn't have the microenvironment in order to allow that, which is not super surprising. But I would never have done the experiment, and I appreciate they did it because it really does show exactly what's going on here, which is they are sort of reinforcing this long-lasting Th1 response in the upper airways.
Yeah, and so now, dear listener, we are getting into the snark-laden portion of the episode, whereby I say, now we have established that we can, at the same time, with a mere 3 series, like, or fourfold series of injections over the course of a month, we can eliminate all respiratory infections and block seasonal allergies at the same time. So where can I line up to get these injections is my question. Because I don't want to get colds anymore. I don't want to get SARS-CoV-2, and I don't want to have allergies. So, like, let's get this through clinical trials. Let's go. What's the problem?
They may. They might. They might. The challenge, and we will see, right? Because this is now, it becomes a question of tuning, right? What we have just seen is that if you highly polarize this environment in a way that is sufficient to drive epigenetic changes and sufficient to, in the long term, prevent allergy because you are so highly polarized, it is not clear what it would feel like as a patient, right? Now, they measure
mice
weight loss.
do not have symptoms.
No, they measure weight loss. So these mice are not catastrophically failing, right? There is an inherent safety profile involved in this. It is not clear how it would feel to have this. People generally report off-target effects of vaccines that are injected, like inflammatory systemic effects, fevers, things like this. Some of that might have shown up in the mouse model if it was going to be a real problem. I imagine, knowing Bali, knowing the quality and translational bent of his group, I imagine that they're doing this now in macaques. I imagine they're looking towards what a human trial would look like. It will be important to see what it actually feels like to have this kind of chronic immune activation stabilized to a point where these tertiary lymphoid structures set themselves up. One can imagine that some of the symptomatology of long COVID, for example, comes from chronic inflammation of the lung environment, and you might accidentally be recapitulating some of the symptoms that people are trying to stay away from. By doing this, and we won't know until we actually.
I think that's a very strong might. I think you don't get anything for free with the immune system, right? There is a reason that we have not evolved to have tertiary lymphoid structures in our upper airway. We are constantly being assaulted by respiratory infections. There's a reason that this is not a physiological response that we typically mount, right? Like, crucially, most of the symptoms of a cold that you get, all of the things that make you miserable about a cold, are responses of your adaptive immune system and your innate immune system trying to clear the infection, right?
Which is why ibuprofen works on everything, by the way. You're not actually doing anything to the infection, really. What you're doing is you're knocking down the symptoms because you're knocking down the immune response, right?
Right. And if this response only lasts for 3 months, what we're talking about is I think the most likely outcome of a human trial of something like this is that you basically have to give yourself a cold every 3 months to avoid getting colds.
Yeah. Now,
right?
Yes, I agree. Here's where I think we
get 4 week long cold every 3 months.
Yeah. Here's where I think we do get into interesting territory. And they mentioned this in the discussion. So, bringing it back to the realm of what would we actually think that this is applicable for. COVID-19 was terrible in that it was a primary response to a pretty bad viral pathogen, not the worst viral pathogen that we've ever seen, but a pretty bad viral pathogen, right? Mm-hmm. It gets worse than that, right? I think that any immunologist will tell you that, we were erring on the side of hoping to prevent deaths at the ends of our physiologic spectrum, right? We were worried about children in the beginning with the MISC. We were rightfully worried about adults, especially those older than 70, because it was very clear that people with susceptible immune systems were going to get hit hard by SARS-CoV-2. And that's where a lot of the public health response came from. It gets worse than that. The 1918 flu was way, way worse than SARS-CoV-2, at least in the middle of that population, right, where you're killing otherwise healthy people.
Well, and more to the point, SARS-CoV-1 was really horrible. It had different infection dynamics, so we managed to contain it. But,
Right, and it gets worse than that too, right? There have been pathogens in human history where, lethality creeps up to like 20, 30%. And we don't, exactly. And we don't have that in our recent memory, but that's a real thing that exists. This is where I think something like this could actually be a really valuable piece of information to have in hand. If you have a respiratory infection that starts to creep up around a 5 to 10% mortality, that is globally catastrophic. If you could immunize somebody, even if it makes them feel like crap, right? And keep people alive for long enough to put together a vaccine that's actually capable of putting that pathogen to bed, or at least providing some backbone immunity, that is a place where I could see this. I think the news covered this in a way that sort of suggested that we were going to cure all upper respiratory infections in a non-specific way. And the answer, I think, Kevin, to your point, is like, maybe you could do that, but it would probably come with an enormous cost that we have not yet seen through this preliminary work. But as a temporary fix, as a way to knock down something that's potentially really, really bad just by putting together a pro-inflammatory, highly activated environment. Because that's basically what we're doing here, right? We are priming the mucosa to highly resist any invasion, right?
Yeah.
That's what we're doing. At least it feels to me like that.
Yeah, it's an interesting idea. I think it feels less convincing from a scientific standpoint, given that we have mRNA vaccines now. Because like the amount of time, the year between sequencing SARS-CoV-2 and having a vaccine go through clinical trials, that year was primarily for regulatory reasons, because we'd never done an mRNA vaccine before, and scaling up the manufacture. The actual formulation of the SARS-CoV-2 vaccine, we had it within like 2 or 3 weeks, right?
2 weeks.
2 weeks. So if we lived in a rational world where people actually understood and were not like irrationally scared of the mRNA vaccine because they worry about, microchips being inserted by Bill Gates or whatever the hell is going on, for any global pandemic potential, we could just scale up mRNA vaccines to get there quickly. Given the world we live in, maybe this is going to be more appealing. Maybe you just like put it in with a little bit of ivermectin, and you can get half the population that's afraid of mRNA vaccines to take this instead. The cold symptoms are because it's working. That said, like, I think the point that you made at the beginning is that it's excellent science. It's really useful immunology, and like the theoretical backbone behind this is really important to understand. I'm really curious. One thing that I wanted to know, and beyond the scope of this paper, it's a follow-up, but like what I want to know is, can you keep doing this? So like I was making a joke that you have to do this every 3 months to maintain protection, but can you? Because by the time you get out to 3 months, now you've got an established adaptive immune response. Your immune system has got the memory. It should be able to just like knock that shit down immediately. And so I wonder if you'd have to keep cycling the antigen if you wanted to do this a second time. Mm-hmm. I think there's a bunch of cool follow-ups to this. It's a cool paper. It's neat. I think my main objection is to the news coverage of it, not to the actual paper itself.
Yeah, no, that's fair. That's fair. And I think that, like you said, it provides a really important context for the conversation around things like BCG vaccine, like polio vaccine. It sort of puts a flag in the ground and says, like, here is actually what this might look like in these cases where it seems like non-specific immunity is having a profound effect. It turns out you can polarize for a long time, and when things polarize, you become resistant to the things you're polarized towards, as well as to allergy.
Yeah.
I love that. I wish they had done a worm infection.
That would have been cool.
I would actually
see if it's more severe.
Exactly. Yeah, see what happens. Or leprosy, where you have to make that type 1, type 2. Like there are some interesting components of that could be explored. Regardless, really interesting work. I enjoyed reading it. Actually, I enjoyed this discussion. Totally. I'm sure that we'll hear more about this because if I know anything about Bali, that we will I think test this in every setting in the best way. I think his model, at least when I was in the lab, was to find something really interesting and then basically put it into every scenario that could be thought of and just see how it responds. And I think that's really reflected in this work. And it leads to a much more broad understanding, I think, of some of the fundamental concepts than had you just like walked down. Because if you can imagine, if we had just stuck with SARS-CoV-2 here, right, as most people would, we wouldn't have seen the actual
100%.
thing. So.
Yeah, totally.
Yep.
Yeah, cool paper. dumb reporting, but cool paper.
That's right. Normal science reporting.
This has been Audio Immunity, episode 53. I'm Kevin Bonham with Matt Woodruff. Like I said, we're on Substack now. Audioimmunity.org, though, is the place to go for all the links, RSS feeds. If you want to support us, you can go on Substack. Right now, the current subscription is only $5 a month, but if you want to do less, send us a message. We can send you a discount code that we'll just send to anybody that wants to support at lower amounts. We're happy to do that. Let's see. We're on YouTube, youtube.com/audioimmunity. We're on Blue Sky. Again, all those links are on the website. Just go there. Like and subscribe on iTunes, though, if you can, or whatever podcatcher service you're using. Share us on social media. Let us know if there's any other topics you want us to cover, or if there's any other science reporting that you want us to slag on. Let's see.
Or promote, sure.
If you want more grousing about scientific review, you're going to get that for like another 2 years before Matt and I both have to shut our labs down.
Yep.
Because we're not getting any grants. Music at the beginning and the end was composed by Rachel Reinick, and we'll see you next time.
Take care, y'all.
Sometimes you get lucky.